All right, so we are going to be talking about genetics or the study of genes and hereditary. Now, I am going to go pretty fast through this video, so it is not 18 minutes long because that was my first take. But we've already looked into genetics a bit, the study of genes and hereditary. Back in Unit 1 with lions, we tried to figure out what parents were the parents of certain cubs. And we used the genotypes to get a more specific answer because the phenotypes didn't leave us with a more conclusive reasoning. So those phenotypes were the physical traits and the genotypes were the separate alleles, okay? We knew one of those alleles came from mom and one of the alleles came from dad. Now we are going to be looking at basic Mendelian genetics. So let's start it. It has been generally accepted for many years that genetic traits of the offspring of any species were the blending of traits presented to the parents evenly, meaning exactly half are from dad, exactly half from mom, so you should look exactly like half of them. That was a general misconception. It was also commonly referred and accepted to that hybrids would revert back to their original form because they didn't know any better about dominant and recessive traits. Because they thought that if you were a hybrid, eventually you would work back to a more purebred form before you were able to reproduce. Now we know that's not true because of things like our apples. Our Granny Smith apples and Red Delicious apples look vastly different from each other, but those two apples can reproduce to create a certain different varieties of apples, and then those varieties of apples can continue to reproduce to continue that that line of that variety of apples. They're still apples, but people thought they were different species at one point, creating hybrids. Not all hybrids are going to be able to reproduce. Some hybrids, like ligers or a lion and tiger mix, they aren't able to reproduce at all. So we aren't going to get too deep into that, but I wanted you to be thinking about it. This was all until an Australian monk named Gregor Mendel in the mid-1800s observed garden peas. His research continued over 8 years and involved tens of thousands of individual pea plants. To this day, he's known as the father of genetics. He used very tedious experiments to be able to breed pea plants, not so that they're able to breed normally like they usually do out in the wild, but he had to actually use a paintbrush and to very tediously move pollen over from one plant to another to breed them. It was a very, very tedious process, and I commend him for his research because he sparked a lot of research for many other avenues. We are going to watch this quick video here, and I'm sorry this is going to take up a bit of time, but I want you to take some notes in that handout that you should be filling out. So the digital version of this handout is those guided notes, so make sure you're taking notes in there. You can always stop the video and go back if you need to, so that you are able to take quality notes.
Most teacher and biologist Gregor Mendel is known today as the father of modern genetics. Gregor Mendel's experiments on sequence were fundamental in establishing the field of modern genetics. Born Johann Mendel on July 22, 1822, in what was Austria and is now the Czech Republic, Mendel excelled at school and became a popular figure in monks to further his education. He was given the name Gregor when he entered the monastery.
The monastery sent him to the University and that gave him the education in practicing math and so broadly that he then brought back to be a teacher.
Mendel accepted a teaching position at the monastery in Brno after completing his university studies. It was during this period he began his famous pea plant experiments. Before Mendel, people thought he was equally inherited with the DNA and DNA in equal proportions.
Mendel experimented with pea plants and found that instead the dominant and recessive traits were all organisms that were passed on statistically based upon the mixing of genes between the parents.
This theory is now known as the law of segregation. Mendel also formulated the law of independent assortment.
The law of independent assortment addressed the fact that there are a large number of traits. These traits are passed on randomly
Proudly Vanilla.
According to Gregor Mendel's research, disregarded it wasn't until the early 1900s, after his death, that Mendel's work was appreciated, and that shows as the basis for genetics and heredity.
In his later years, Mendel became the abbot of his monastery and spent less time in scientific endeavors. Gregor Mendel died in 1884 at the age of 62.
Before Mendel's work laid the foundation of modern genetics, modern gene therapy, and is the basis for some of the most exciting discoveries in medicine currently and in the future.
All right. So again, Gregor Mendel, he laid the foundation for pretty much all of our genetic research that we currently have to this day, including modern medicine. So moving on to his law of segregation, through Mendel's findings, he discovered that there's much more at play than simply the blending of traits. He was able to track genes and their appearances in offspring and be able to view now dominant and recessive traits. So we know that genes come in pairs, one's from mom, one's from dad. Now, whatever one that we typically look like is a blending of those traits, and a dominant trait will typically go over a recessive trait. The dominant traits being things like brown eye color will go over things like blue eye color, which are a recessive trait. So you have more of a chance to have blue eyes typically from parents who have brown eyes and another parent who has blue eyes. You'll typically more likely have brown eyes in that case because brown is a dominant trait. It dominates over those recessive ones. So he studied, I have to delete this. He studied purebred plants of opposite phenotypes. The particular ones he did study were short versus tall plants. But we are going to be looking at different crosses of different varieties of plants. So a cross is a specific organism, a mixing of specific organisms to test and see how traits are inherited. So a cross, I like to think of one plant goes on top, this is the mom plant, this is the dad plant, and then they are crossed, and so it looks like a cross. Now there's two main types of crosses. There's a monohybrid cross and a dihybrid cross, mono being one trait and di being two traits. So in this case, we have a yellow pea pod right here, and we have a green pea pod right here. Now in this cross, the yellow pea pod is going to be recessive, representing with a lowercase g, whereas the dominant pea pod is going to be a uppercase G or a dominant trait. When it's crossed over, the big G's or the dominant traits go across first. So I put a big G in the boxes that they are next to. And then the little g's come down. The recessive traits now come down and go in front of the big G because the uppercase letters always go first. I always try to think of that. So big G, little g, big G, little g. All of these are still going to be green pea pods because that G, that green color, is dominant over the yellow color. Now in the dihybrid cross, we have more varieties going on here. Now we have round versus yellow plants. So these are talking about peas itself, not the pods. So the dominant ones are going to be round and yellow. So in this case, we have a dominant round yellow, a dominant round, a lowercase y, which represents green, and a lowercase r that represents wrinkled. An uppercase Y is the yellow, and then wrinkled and green. All right, so we have that variety as the mom, and we also have it as the dad along the side here. Now, as they cross, we bring the dominant traits over first. So dominant R, and then dominant R, dominant Y, and then dominant Y. So big R, big R, big R, big Y, big Y. So this plant is going to be a purebred round yellow plant. But purebred doesn't really matter. It matters what traits are going to be expressed in this case. So that's why there are 9 separate boxes out of the 16 here that are going to be round and yellow. I'm going to point out a few of these, and I want you to be able to point out the other ones. So right here, we have a big R, a big Y, and a little y. Now knowing that that big Y is dominant, it dominates over the little y, which is the green color, meaning this is going to be yellow. Okay, so a round yellow, meaning that it's going to fall within that 9 of those round yellow phenotypes. So the big R, the big R is still going to dominate over the little r, and then the big Y, meaning it's still going to be a round yellow phenotype. So I want you to be able to pull out those different varieties within this Punnett square, which is what these are called. They're called Punnett squares. Now, Mendel looked at tall versus short plants. So he took purebred tall plants and purebred short plants. Now, he knew they were purebred because he crossed purebreds over and over and over again until they only produced tall plants. Same thing with short. He crossed short plants over and over and over again until he knew for sure that they were purebred short plants. Now, when he crossed them together, these created offspring that were all tall. So that big T, he knew that was a dominant trait, and that little t, or the short, was going to be a recessive trait. Now, once those plants crossed with each other, he noticed that 3 of them were tall and 1 of them was short, meaning there was a 25% chance that they were going to produce short offspring. Now, they had a 75% chance they were going to produce tall offspring still. Okay, 2 of these are purebred. 1 is short purebred and 1 is tall purebred. And then the hybrids in the middle are going to have a heterozygous genotype, meaning they have 1 dominant allele and 1 recessive allele. Okay, we know 1 of these came from mom and 1 of these came from dad. Now, this is played out in not only just tall and short, it works in humans as well, but also in eye color. So we have different genotypes, which are the individual letters here. Those are the genotypes. And then the phenotypes are going to be the physical representation of these letters, okay, the physical look. Same thing with eye color. That's going to be the physical look. Whereas if it's letters, like we saw in back in Unit 1 Lions, that is the genotype.